All right, thanks for coming, everybody. Thanks to the organizer of the Biophysical Techniques lecture series. Thank you for everything. Thanks for inviting me. This is Fabrice Gorrec, responsible for the Protein Crystallization Facility at the MRC Laboratory of Molecular Biology. The facility, part of Structural Studies Division, is open to all LMB scientists. It is also open to external collaboration after agreeing on fundamental aspects, especially ethical and publishing aspects. Despite the spectacular technological advances in cryo-EM and protein structure prediction algorithms, X-ray crystallography remains a powerful technique in drug discovery, enabling us to determine highly accurate structures of small and medium-sized proteins, protein consisting of several dozens to hundreds of amino acids, let's say. This with small molecule ligands, although I'm not going to cover working with compounds today. This graph shows the number of crystal structures released annually in the PDB over the last 3 decades. The total number of entries is getting close to 200,000. They include structures of protein-protein complexes, protein complex with RNA, DNA, and oligosaccharides, and also structures of nucleic acids on their own. There is a steady pace of 8,000 to 10,000 structures a year over the last decade. The vast majority of structures are solved under 3.5 angstrom resolution, which is what we want for coherent and trustful data. Half has a resolution below 2 angstrom. The average molecular weight of the proteins is around 90 kilodalton. By comparison, the average molecular weight of structure determined by cryo-EM is 10 times larger. Macroscopic protein crystals are assessed under a light microscope. Single 3D crystals with sharp edges and symmetry, like the one shown here, are fished out with microloops for X-ray diffraction tests. The integrated diffraction spot's intensity is directly proportional to the volume of crystal illuminated, and hence crystals must have dimensions of at least 20 to 30 micrometer, even larger when testing them with our in-house X-ray diffractometer. We have a Rigaku FRE Plus super bright X-ray generator at the LMB, the most powerful in-house generator commercially available. It is set with 2 MAR-DTB detectors. However, we most often proceed with remotely after shipping frozen crystal to the diamond light source in Oxford. All this is dealt by Dom, our expert crystallographer and X-ray facility manager. Appearances can be deceiving when assessing protein crystals. The diffraction pattern is the only data that matter along the process, as it shows the true intrinsic qualities of a crystal, notably the high level of order within the crystal lattice. This is the kind of diffraction pattern we want to obtain, with high density of sharp and single spot at high resolution. Quality diffraction protein crystals are typically hard to produce. The underlying reason is the multitude of parameters associated with the nature of protein samples, their crystallization, and the resulting crystals. Here are the main factors affecting crystallization, and quite a few, as you can see. So we're not going to cover all of them today. These are splits in 3 categories: physical, chemical, biochemical. Testing all these factors represents a surrealistic effort. Besides, they are mostly dependent to each other. Surely, the amount of sample is limiting anyway. A way to alter many variables at once is to screen the sample to be crystallized against a relatively large number of crystallization conditions that are formulated with a broad variety of reagents, such as precipitating agents, buffers, and many more. That's how we proceed at the LMB, our main strategy for initial screening. Here are examples when useful crystals were obtained during initial crystallization screening at the LMB against hundreds of conditions. The corresponding yield is a bit less than 1%. Considering other cases where no useful crystals are obtained, our overall yield working with novel samples is estimated to be far below 1%. Crystallization is usually studied according to two main steps, nucleation and growth. Cryo-EM is increasingly used to better understand nucleation. Observation using time-resolved cryotransmission electron microscopy suggests that nucleation of glucose isomerase progresses through specific interaction between only a few protein molecules at a very early stage of nucleation. Before that, the theory was more like people were talking about more than global. There was this kind of aspect where the specificity of interaction were not seen at the beginning. But these groundbreaking works from the University of Brussels show that a main mechanism of nucleation is polymorphism selection that takes place at the earliest stages of nucleation. We could say it is based on specific building blocks. In fact, only a slight difference in the precipitant concentration means glucose isomerase can crystallize into at least two different space groups or can aggregate into a disordered gel state. It was shown with atomic force microscopy that multiple nuclei can appear on the surface of the crystal and become two-dimensional patches. This explains a main mechanism of crystal growth by sequential layers addition, as two-dimensional patches go tangentially and merge to form a layer. So because there are several of 2D growth around the surface of the crystal, this is a constraint, and it's continuously creating step edges on the growing surfaces, which can lead to dislocation between spiral structures, and that turns to crystal imperfection. The lattice of a micrometer scale protein crystal is formed with hundreds of millions of unit cells. That's not the slide. Sorry. Here we go. Sorry. The lattice of micrometer scale protein crystal is formed with hundreds of millions of unit cells. The proteins hold together through weak interactions between small hydrophobic surfaces, longer range and flexible electrostatic interactions as well, and sometimes even a single hydrogen bond. In addition, the density is typically very low, with 50% solvent content or much more. This plasticity, not only limits growth, it leads to structure defects. A common issue among crystal lattices is mosaicity, a poor alignment between the unit cells. Mosaicity broadens the diffracted beams and lowers the signal-to-noise ratio on the detector. Another crystal pathology occurs when crystals are made of two or more lattices in different orientations, which can prevent the solid structure. Shown here, a most straightforward example of twinning where a crystal is formed with two pyramid structures in opposite orientations. But that's not going to make the data processing easier. So it's often said that the sample is the main variable of protein crystallization. There is a plethora of cloning, expression, engineering, and purification systems that can be tested for producing high-quality crystals. In principle, rigidifying and shortening samples increase the chances of successful crystallization, but not systematically. Other directions may be taken. The case of inherited congenital cataract illustrates well the possible effect of protein sequence alteration. A single point mutation causes the crystallization of one of the main protein of the length, the human gamma D crystalline. Single mutation is, however, not a starting point when trying to crystallize a novel sample in the lab. It is something investigated at later stage for crystal optimization and discovery purposes. To increase the chances of yielding useful crystals, outermost disordered region of the protein surface should be removed, the so-called floppy loops. The limited proteolysis method is based on short and controlled exposures of the protein to proteolytic enzymes. You will need the support of our mass spec facility led by Caterina Franco to deduce the shortened sequence of interest. To bypass this time-consuming process, one may try to integrate traces of proteases directly into crystallization experiments. We have the required kits in-house for this. You will need the support of an X-ray facility manager for this. Don't believe me. Some useful protein engineering strategies are based on enlarging the protein and increasing the complexity of the sample. On the left, a T4 lysozyme sequence that crystallizes readily have been fused on the N terminal of a beta-2 adrenergic receptor to obtain useful crystals. The lysozyme provided additional surface area favorable to crystal lattice formation. What you do here basically is you're varying the structure you have to change the possible combination of interaction and then generate a crystal that wouldn't occur otherwise. There's also another strategy for this, is to use an antibody fragment as chaperone, like you can see on the right here, this to stabilize the protein and also facilitate crystallization. So hopefully you don't want to reach that stage, but because it's lots more work in the process in parallel. But sometimes if you really have, it's impossible to stabilize your protein, this is definitely an approach you want to try. Protein stability is key to successful crystallization. A thermal shift assay, more thoroughly presented in another talk of this series, is a relatively straightforward option to identify stabilizing agents. Essentially, an environmentally sensitive fluorescent dye to track the thermal denaturation of protein samples. Proteins generally increases dye fluorescence as their hydrophobic cores become exposed during denaturation. This is not suitable for all proteins, though. And to know more about this, you need to discuss further with Stefan, which is here today, and head of the biophysics facility. But there are many more things to consider when things doesn't work. How are you going to alter your sample? There's the post-translational modification, of course. Maybe you need to change expression system here too. It may end up with lots more work. You have to re-optimize everything. But that's definitely something to try if you reach a dead end. It may be simpler to, for example, to remove I'm not going to say right, decoagulate the sample, to remove the sugar chains on the surface of your protein, which may be on the way of crystal contact. You may find a method with some enzyme or chemicals that can be useful instead of reconsidering the whole expression. Purification, are you fully in control of the conformation state? So it's not always obvious the proportion of your protein in the right conformation, and the different population in conformation is against crystallization, obviously. Are you also in full control of the oligomerization of your protein? A major issue for a lot of protein is polymerization. So you want to try to find and alter the polymerization site. That's what people of Jan's Love group are doing very often. So if you have questions about that, if you have polymerization problem, I guess you're better off asking members of this group. It represents a lot of their work when they want to crystallize something. And there's also the cofactors. How do you work with cofactors? That's the question. What's really needed to have the cofactor there where it should be. Maybe there's biophysics assays previously to be done. Which one as well? If you I see people putting ATP half-life is what, 1 picosecond. So there are chemicals like non-hydrolyzable ATP, that you can use. And maybe also there are cofactors that are not in the literature yet. They are yet to be discovered. So you may want to look at that as well and try your luck with different cofactors. Here is a 2D protein solubility phase diagram where saturation levels are represented according to the 2 main parameters that alter the protein solubility. Because crystallization occurs at supersaturation. The sample and precipitant concentration are the two parameters here. The solubility boundary curve divides the diagram into two areas, undersaturated and supersaturated. The supersaturated area comprises three zones: metastable, nucleation, and precipitation zones. So that's how we represent the phase diagram for protein crystallization. This is also a very standard way to show this with only two parameters. You may be able to make one yourself with proteins with very stable, small globular protein, but it may also look very different, or it may not be able to do this kind of phase diagram. But that's the kind of diagram we show as a basis to understand the supersaturation and the different zone. In practice, many other parameters should be considered. For example, transition from aggregation to crystal growth is regularly observed for large and complex samples. Also, the oligomeric state of a sample may change with the saturation level and the pH. And I remember seeing it's very rare to have phase diagrams, but I remember seeing one with that actually showing that different oligomeric states of the protein. Protein stability varies also with other parameters like the surface tensions, etc. Growth preferentially occurs in the metastable zone at lower saturation level than nucleation. This is a bit in contradiction with all the crystallization methods that are based on steadily increasing the saturation. Basically, when you're increasing the saturation, you want to reach the precipitation zone where you have more chance to have nucleation. But then the growth of the crystals is occurring in a better way in the lower level of supersaturation, while your experiments is still, going higher, triggering more and more saturation. So this is a limitation, which is the basis of, this limitation is the basis of a method called seeding that I'm not going to discuss today. There are different interesting and useful crystallization methods. Vapor diffusion with seating drops is currently the most widely employed technique, mostly for practical reasons. I'm going to talk only about this method today. Setting up vapor diffusion experiments starts with mixing the protein sample and the conditions to form a drop that sits next to a reservoir, as shown on the figure here. The reservoir contains a much larger volume of the same crystallization condition. The experiment is swiftly sealed airtight, allowing equilibration of the osmolarity to take place between the sample containing crystallization drop and the reservoir. Because if you see in the reservoir, you have lots higher concentration of the precipitating agent. It could be a salt like ammonium sulfate, or it could be a polyethylene glycol. So basically, this will pull out, so to speak, the water out of the drop. And all the concentration of all the components in the drops are going to increase. The mosquito technology relies on microsyringes for positive displacement pipetting. So we can work with some of the most viscous conditions, because concentration with high concentration of pegs or glycerol, for example, we get quite a few of those. And just to mention as well that if you want to set up a plate screen, which is 96 condition, for the 96 droplets working 100 nanometer scale, you need 16 microliter of sample. If you were doing the full screen to investigate thoroughly your sample at the first time, especially if it's a novel sample and you don't have a clue what are the important reagents there or how it's going to work, then you may want to do the full screen. And in that case, if you do the full screen, 24 screens and 24 times 96, that's 24 times 16, sorry, that's 384 microliters. We have a mosquito in the cold room as well to operate. So obviously now, remember parameters, if you want to switch completely, the parameters then work at 4 degrees, but I'm not going to talk about that today, but it's possible to set up plates and store them at 4 degrees here at the LMB. Screen formulation. So the crystallization reagents are typically classified as precipitants, buffers, and additives. The combinations of these reagents alter the combinations of variables associated with the main parameters of crystallization. There are different approaches to formulate screens with the multitude of suitable reagents to be tested. The small green cubes shown here represent combinations of 3 components in a conceptual space for 3 main types of formulation. A grid screen, left, consists in a systematic approach to screen formulation. So you have 3 parameters, and you basically very rationally varying them. Obviously, you can't reach, that many chemical space. You can't do a very narrow range of concentration. You can't test that many reagents. So there's a limit to a grid screens. Because of the large number of reagents well suited for crystallization, an extensive grid screen would include millions of combinations, in fact. So it's technically not feasible. Again, that will represent a surrealistic effort, but also, in any case, you have a limited amount of sample in general. So the most developments of initial screens are based upon minimizing the number of initial conditions, such as with the incomplete factorial and the sparse matrix formulation. An incomplete factorial, the one represented the same way as the grid screen in the middle, could be defined to some extent as an extended grid screen with combination omitted randomly. So there are different ways to do this, but basically, you want to expand the grid screen somehow. For that, you just either you miss some components or you miss some concentrations, but you just find a way to do that. The sparse matrix screen, the one represented on the right, that's simpler in a way. It's just a selection of conditions that were successful in the past that you put together with more. So either you're trying to covering a huge set of results or a smaller subset of results, for example, for a certain type of proteins. Again, there are different ways to do this, but basically that's the idea. So I did this slide because people often ask about assessing the experiment, because this is also a bit a tedious task, especially if you want to cover a lot of conditions and you do a lot of optimization. So basically, and we're talking obviously still vapor diffusion and at room temperature. There's no strict data available about this, and it will, as for everything, depends on the samples. But in general, what I see is that, so when you start your vapor diffusion experiment, which is mixing the protein sample and the condition, the first hour, you may have liquid diffusion happening and then the subsequent hits, because we don't mix, it's too small to mix the drops. The drops are too small, so we don't mix them. So I realized that there's first, for some of them, there's a liquid diffusion occurring first in the drop, and that actually can produce heat. So you have very early heat sometimes. It's a good time as well to check if you don't have very false positives which are there, which were just dropped there. For example, some salt that were not well solubilized from the reagents or things like that. There could be also very tiny contaminants, like very tiny pieces of plastic that look like crystals. So you will see that as well at the very beginning. After a few days, 2-4 days, you have most of the equilibrations between the drops and the reservoir are completed or almost completed. So at this stage, it's a good, if you don't want to check your plates, too many times, that's a good step. So let's say you wait 4 days, and then you have a first results that's, is giving you a good indication if it's going to work or not. Then I would say after a couple of weeks, now all the vapor diffusion experiments are completed. So it's another good timing to check. After that, it's a bit more vague. So after months, depending also on the quality of the plate, the quality of the seal, the one we use, the MRC plate with the seal we use is very good at that. The dehydration is not occurring before months and months. But let's say after 3-4 months, you have what I call eventually the dehydration hits. So now you're not looking at what's happening with the vapor diffusion experiment, but with the overall dehydration of the plates. So you further increase the concentration. There is a way to do that, which I forgot to put in the talk, which is something that Dom developed, which is putting some highly concentrated formate in the reservoir to accelerate this step, actually. I need to put that next time. And then, again, after that, it's quite vague to control things. I mean, what data is there? But some people like to do a very last check, a year after. And it's true. So there are some surprises sometimes. It's hard to explain one. Again, more dehydration, or it could be also a long-term rearrangement. The weakest molecules, are gone, and you're left with some rearrangement for some whatever ways that's beneficial. There are all those theories about this, but it's quite unclear. But it's not a bad idea, basically, to check your old plate one last time before you chunk them. You may see, however, that a lot of drops are just simply dried out. So that's what you want to see. That's if you're lucky. I call it the phase diagram droplet. Sorry. This is work from Marc Fillier. What's interesting here, and that makes it very clear that it's a protein crystal heat, is that it's actually, you can see in a way the representation of the phase diagram with precipitant, nucleation spots, average size crystal, larger size crystal. I suspect that this could be the result of liquid-liquid diffusion instead of vapor diffusion, but I'm not sure. But that's definitely the kind of hit you want to see. Unfortunately, the more commonly observed results is more about precipitant. And there's lots more type of precipitant, some more or less interesting that you may see in the drop, like maybe you are at the edge of nucleation. You see lots more about that on the website of Therese Bergfors, which I would advise you to go and see, because she's explaining all of this very well. About precipitates, what does that mean? Well, it can mean a lot of things. Some people also can go a bit crazy with this. For me, this just means that the protein precipitates against one of the reagent in the conditions. That's what it means. So it may be meaningful if all your drops are precipitated, in which case you can conclude that your protein is basically not stable enough because it crashes with every possible reagents. There's a problem there. It may mean it's too concentrated. I'm not sure I like this notion of too concentrated, but it could mean that too. The other way around, if all the drops are clear, there too, you may put some meaning on this. Maybe you don't have proteins there. That's a possibility. Or it's very low concentration. Or it's too stable in a way. It's funny to say that, but it's too stable in the sense that you can probably push the concentration lots further than you think with it. So some acknowledgement here. I wanted to thank my manager and Olga, Jan and Olga as well, which are the founders of the protein crystallization facility. So everything we do today, they've set it up a long time ago now. And then I arrived as a support scientist to take care of the facility and learn everything with them. Dom Bellini as well, which is not doing only crystallography. So I thought I would call him the everything everywhere all at once man. Because he's a bit everywhere. And also running the facility, it's support, some aspects, you need support for that nowadays where everything is becoming a bit more complicated. And I need to thank the people of the operation groups as well, Christine and Tom, which solve IT problems showing up regularly, and Poppy and Laura from procurement and estates. So it's good. Because I added a EndNote slide, which are the essential consideration about the sample, which is now a very practical thing that people wonder usually if they don't have experiences and then arrive at the facility. So I thought I will actually finish with that. This in a way that's what's most users and new users want. Molecular weight. We want ambitious structure. So you want to aim for large things. But basically, this rule of thumb is the success rate, the yield of crystal is inversely proportional to the size of the protein. But again, I like to try something, we want to try ambitious things. So don't go straight away to try to chop it to have more chance. But sometimes you have to. Maybe you can work only with the active domain. So in which case you have, the smallest bit possible of your protein. Purity. Please don't come to the facility with, something you're not sure what it is and how pure it is. Single band on your gel, please. You've done your homework there. Obviously, that gives you more chance. Maybe you can't purify thoroughly, and then you did your homework and you work hard on it, and then at that stage, then maybe, if your gel doesn't show something that great, that's all you can get. Again, be ambitious and try anyway. The crystallization is exclusive process, so there are cases where you actually have different proteins, but the one that were in higher concentration and the most stable crystallize, excluding everything else. We talked about that a bit earlier: stability, conformation, oligomerization, polymerization. This is key factors that you need to look at as well to have more chances to crystallize your sample. A very basic question that we have all the time: what concentration? Well, that will again depends on the sample, what you can do, and also what is the right concentration for it to crystallize. This is also empirical. If it's very hard to produce amount of sample, you can have a go with 2 mg per ml. That would be the minimum, I think. Under that, there's no chance to crystallize it, even with conditions that are highly saturated, or it will be very rare that it does. But let's say that 5 to 10 mg is a better start to have a chance to crystallize it. Now, 5 to 10 mg, if you didn't shake the solubility of the sample, and in fact, your sample, like the case I was talking about, is very stable, it may be able to reach 100, 200 mg per ml even, in which case, if you start at 5 mg per ml for your crystallization experiments, you probably also far off when you're thinking about reaching the super saturation states. So you may want to look at that, at the solubility of your sample. What I was doing before was I was sacrificing a part of my prep to push the concentration further and further and see what was the limits. And also simple things like testing the effect of desalting the sample. If you arrive as well with half molar sodium chloride in your sample, it's the same. It most likely won't work. So you want to see what's happening when you dissolve. So maybe it will be less soluble, but then it's a balance on solubility and other factors. So I like to start with sample with a relatively low proportion of salt. It's also to have more chances that the impact of all the different vapor diffusion experiments is more there to start with, because if it's highly concentrated, you remember the experiment with the drops, and the water is pulled out in this equilibrium. If the drop is highly concentrated in salts, you won't have this equilibrium to start with. And there are also reasons why a highly salty salt sample is not going to crystallize, but that's one main reason. Another simple thing, the effect of glycerol. A bit of glycerol can make a big difference. Too much glycerol It's a bit the same problem as too much salt. But if you didn't have glycerol, you could run in parallel a sample with and without glycerol, let's say at 10%. A big factor, which is also relatively easy to check first, is the pH. So we have a pH screen actually, so you could just set up a plate with only buffers and look at it as the same way as you look at drops for crystallization to see where your protein is happy or not. That's a simple way to check different buffers. A big one is also integrity after freezing. So the first thing I will ask before someone do optimization, did you do your initial, do you have your initial hits with a fresh sample, and now you're working with frozen sample because you keep some to do your optimization later. You may have surprises here. In general, basically freezing a sample doesn't help. Sometimes it doesn't make any difference, and that's great because you can work with frozen sample, you can have something, do something 3 weeks later, but sometimes it makes a difference. So you do want to have a go with freshly produced sample anyway, and then see if you can work with frozen sample. Again, just a little reminder, if you go to the platform, the crystallization facility, you need 16 microliter at least to do 1 plate. And that's it. This concludes the presentation. Thank you.